majority of stakeholders, and I'm talking here also majority of investors, when they hear cell therapy, they automatically think about CAR-T. And this was one of the first challenges really upfront, delivering the message that we are not doing this. We are not aiming as those price tags. We are having other type of cells, and the process is much, much simpler. And this was one of the first struggles that we needed to overcome in order to really make people pay attention to what we are doing.
Hey, smart scientists. Welcome to another episode on the Smart Biotech Scientist podcast. Today, we are talking about translating scientific breakthroughs into clinical therapies. And to do that, I brought Deana Mohr into the Smart Biotech Scientist studios. She is the CEO and co-founder of MUVON Therapeutics, a clinical stage spin-off from the University of Zurich. So we are in for a conversation on the development of a novel regenerative therapy platform for the treatment of diseases related to skeletal muscle damage or degradation. Stay tuned for an exciting episode. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Deana, on the Smart Biotech Scientist podcast.
Thanks, David, for having me here. Pleasure.
It's a huge pleasure to have you on today. Deana, share something that you believe about bioprocess development that most people disagree with.
Well, I think that bioprocess development could be quite different depending on the cell type that you are trying to understand. And muscle cells cannot be monitored, controlled, and modeled like the suspension cells, like NK cells and T cells. So it's quite a different mechanism there. And most people think that cells are cells, which is really not the case, starting from the big difference that the ones are adherent and the others are free-floating.
Yeah, absolutely. Cells are much more complex than we think, and we're going to get into much more detail later on. Before we do that, Deana, tell us how you got started in biotech and how did you get to your current role as a CEO?
This started actually by a lucky chance, I would say. I was working on my PhD thesis at ETH on a project on regenerative medicine and stem cell tracking for muscle regeneration. And then the results of the thesis worked out, showing safety, feasibility, and efficacy of this tissue engineering therapy. And we submitted the whole package then to the Swiss medical authorities in Switzerland. For good or bad, they gave us approval for starting first in human trials just a couple of weeks before my defense. So I decided to drop the 2 job offers in pharma and dive in the unknown waters of biotech startup and see these results being translated to a real therapy for helping patients.
Wow, that's exciting. How fascinating is that? And tell us the story now behind your company, because you are heading a clinical life science spin-off from the University of Zurich, and you are developing a novel regenerative therapy platform for the treatment of diseases related to muscle damage or degradation. So what is this all about, and what is your vision and your mission?
So our therapy is really using the power of cells and persons also, it's autologous, for regenerating damaged muscle tissue. What we realized was that skeletal muscle is 40% of our body mass. You can call it one of the largest organs if you want so. And as we age, the regenerative capacity of this organ decreases. When we hit 40, we start losing 1% of muscle mass per year. So this is on one hand due to the aging processes which have an impact on the cells. And there are quite a lot of cell populations in the skeletal muscle environment that are responsible for enabling regeneration. And the regenerative capacity also decreases with age because the quality and the number of those cells decreases. So we thought, okay, but then let's make a booster if this is the problem, and started really working on the optimal methods to find those cells, to extract them from the tissue, and then to be able to multiply them to hundreds of millions to enable this tissue engineering to restore the muscle function of, at the moment, small but vital muscles that sustain quality of life of the patients.
And what disease are you currently addressing?
This is a disease that no one likes to talk about, yet it affects every fourth woman after childbirth, and it's called stress urinary incontinence. It is basically by increased abdominal pressure, the muscles of the pelvic floor cannot hold of the urine, and then you lose unintentionally urine. And we found that often due to pregnancy and also through vaginal childbirth, and also with increased body mass index, these are the most three common factors, it is that the musculature there gets weakened because it's kind of used up. And one of the muscles that is very much important for the proper functioning of the whole urination process is the sphincter muscle that is holding the bladder closed. And we could select patients where the problem is mainly due to this sphincter deficiency, and then brought our R&D project into a GMP-compliant setting, and now we are treating patients by regenerating the force of their muscle so that they can have a happy life again.
Wow, that's fantastic. This is such an important mission. And for those of our listeners who are not so familiar with cell therapy, can you give us just a high-level view of how do you treat these patients?
How it works is that we obtain a small muscle biopsy from the patient, from a healthy muscle, and then we isolate the special population of muscle precursor cells, and then we make from the couple of cells that we get several hundreds of millions of those cells under strictly regulated GMP, this is good manufacturing practice, clean room conditions until we reach the number that we need for the therapeutic effect. And afterwards, the cells are prepared for our tissue engineered final product, and then they are injected really minimally invasively into the sphincter muscle of the patient. All procedures, both the biopsy and the injections, are 20 to 30 minutes in and out, really easy, non-complicated. And the big benefit, which is also differentiating us from the other cell therapies that are known, because the majority is CAR-T out there, is that our patients are not dying. So we can schedule the treatment really easily, and the process does not need any immune suppression or whatsoever. As I mentioned, it's 2 outpatient procedures, very quick, and respectively, we have no recruitment issues for our trials. We rather cannot come up with the queue that is piling up.
And is that a one-time treatment, or do they have to come in a few times until it's healed?
If biology is right, it should be a one-time treatment, and this is what we are aiming to understand with our long-term follow-up study, where we are really then tracking the patients for a period of 5 years so that we can understand also the long-term effect. But theoretically, once the cells get integrated into the tissue, they are terminally differentiated and they cannot go back from fibers to cells. So they are there and they are contracting.
Now, coming up with a scientific breakthrough is one thing. There is a challenge by itself, but now actually taking this and bringing this breakthrough, this therapy to patients is another story. And I've had a lot of conversations on this podcast and with other people in the industry. And there is a common thread. There are a lot of people actually struggling with cutting edge science or technology from the lab into the clinics. So can you tell us, Deana, a bit, what are the specific challenges you encountered while bringing this technology from your PhD into now a company and getting into patients?
This is an excellent question, I should say, because all these challenges are very common, and it would have been great if someone has made the efforts to enlighten the new entrepreneurs in those and to really not adjust timelines that often. So the first thing is the timing. If you think that for a tech transfer process from one CDMO to another, if you want to change it, it takes 9 to 12 months with a highly professional network of experts and with systems that are in place. We were very courageous to believe that we can make it in 6 months from an R&D to a clean room settings. And we were 2.5 people doing this. We managed this in approximately 2.5 years, which is also quite brilliant looking now backwards in time, because we were doing this for the first time. We were supported by more experienced quality personnel from the Zurich accelerator, where we are incubated now for the phase 2 clinical trial. But it was quite a steep learning curve, I would say. And many things which are in the R&D lab clear, you have to write 10-page novel about it in the GMP setting. And I will never forget one of the first discussions that we had on the risk analysis, where we were discussing, okay, what is hand-warm medium? And then we were saying, yeah, hand-warm medium, it is this. And then the quality, no, your hands are colder in winter, they are warmer in the summer, then depends on the room temperature or how nervous you are. There might be different variations between the people's perception, so we have to define it and to measure it. And this is the moment where I realize, okay, this won't happen in 6 months, where we are discussing 2 hours about hand-warm medium. So I think the reality check and the expectations were quite striking at the beginning, but we managed to learn on the way and to make things happen. So I think this translation is taking much more time and is much more complex than one might think. Starting also with all the materials that you use in the R&D are most probably not GMP grade, because it just costs 4 times lesser. Yet in the GMP, you need to have the respective quality grade, and this is then needs to be validated for each and every substance that you use. And it's a lot of documentation, a lot of conversations, a lot of paperwork that needs to be done. So I think this was the first big challenge. Then we needed, of course, to validate the process so that it is for sure 100% working, and you get the quality and the number of the cells that you need at the end. Also in the clean room with the, I would call, new GMP grade materials, which was also time consuming because you have to make it one after the other. Then you have to have your personnel also mindset change, I would say, because in the R&D lab, you go with your lab coat and hand gloves, and then you do your experiment and don't need to fill out 10 pages of documents before you start your experiment. Whereas in the GMP, there is these media field trainings. They need to be really educated to work in highly sterile conditions. Everything needs to be very aseptic, and that they can perform the experiments so that they are assuring the safety and the quality of the product that is then finally injected into the patient. And once this is done, we started asking ourselves questions, okay, but we need to make this happen at scale. Then another black box was opened, which we're tackling, I think, pretty well so far. But then we started also understanding more these critical process parameters of the whole therapy development that we need to pay attention to, others not, and how to make the optimal automated system at the end. So it was, in a short time, very steep learning curve with a lot of things that we needed to take account of. And the future will tell what is coming next.
Yes, smart biotech scientists. Deana is making an excellent point. Heed this advice. Tech transfer is much more complex than you would think, and start very early. And you also mentioned that you need the right experts around yourself just to make sure that you're making the right decisions early on. And it takes more time than you might think. So I would like to talk about just the specificity of your cell therapy, especially autologous cell therapy, because obviously cell therapy is quite a large field. What are the specific scientific and technical challenges or unique considerations you have to take into account when you're developing an autologous cell therapy?
So the first big difference that we encounter also when communicating what we are doing is that majority of stakeholders, and I'm talking here also majority of investors, when they hear cell therapy, they automatically think about CAR-T. And this was one of the first challenges, really upfront, delivering the message that we are not doing this. We are not aiming as those price tags. We are having other type of cells, and the process is much, much simpler. And this was one of the first struggles that we needed to overcome in order to really make people pay attention to what we are doing. And the biggest differences is, first, with the adherent cells, we have very few amount of starting material. So it's really a small biopsy, and you isolate really a few cells that you kick off, and this initial part is very sensitive to be really made robust. Whereas with the immune cell therapies, I would call them, you start with apheresis, and then the isolation is you get big amounts of cells from the beginning, despite being more invasive for the patients. So this is the first main difference, that we start from a tissue and not from blood, and that our cells need some surface to attach and to grow and to fulfill their functionality, whereas the CAR-Ts do not. However, we do not do any genetic modification, so it's much, much simpler process, and we don't have there any safety concerns because it's just making hundreds of millions of them. And I think this is also one of the points what makes patients feel very comfortable with our therapy, because it's their cells, we don't mess up with their genes, and it's considered acceptable for them. And I think the tricky part is now how to make those hundreds of millions of cells, but making these in a short time. Because for every cell-based therapy, the longer the cells are outside of the body, the more you don't know what is happening. So we try really to expand them very fast at a very high quality and to give them back to the patient as soon as possible. So these are more muscle specific.
And what is the difference? Because you have different donors, you have different patients, and obviously our cells behave differently. Are there huge differences, or once you have a process, you can just copy paste?
I would say in about 95%, yes, it is the same process. What is the difference between the different patients? What we are also collecting now data during the clinical studies is that we see, okay, the younger the patient, the faster the cells do the job. And for elderly patients, sometimes you need, for example, 1 week more time to reach the number and then to be treated. It's rather timing. That's why I said it's this variability of the different patients. And what we still don't know is the effect of different comorbidities of the patient. So we excluded those which we were expecting and are expecting to have a negative impact on the cell quality, on their muscle regeneration. However, there are millions of diseases and comorbidities that we could not exclude everything that might have an effect. So this is, I think, one also very important point which we are trying to understand now as we move forward into the clinical development is if some patients respond lesser well or super well, what is their, if you want to call it, genetic background, or what is making it be super good or not that good. So the patient variability is there.
Hey, smart biotech scientists. I hope you've been enjoying today's conversation with Deana Mohr. Deana is the CEO and co-founder of MUVON Therapeutics, a clinical stage spin-off from the University of Zurich in Switzerland. What did you think about today's episode? Please stop now and leave a review on the platform you found us on. I look forward to hearing from you and hearing what you liked about today's episode. And perhaps you are navigating the transition from R&D into the clinical studies and process development as well. And you're not so sure what you should do and how you should do it best. If this is the case, I want to offer you a free assessment to get started. So book a call with us so we can help you translate your breakthrough science into clinical therapies. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.